Okay, so we are recording now.
Excellent. Okay, everyone, welcome to today's seminar. I'm Mark Giltnan, professor in the Plant Science Department, and I'm really pleased to introduce today's speaker, Noah Winters. Noah has an interesting background. He has a Bachelor's of Arts degree from Kenyon College, and he enrolled in the ecology graduate program at Penn State about 4 years ago. He's in his fourth year now. One of Noah's really, he had very good timing. He came into Penn State at a time when my lab, along with a couple of other co-PIs, had just received an NSF project, a very interdisciplinary project. And so Noah actually came in with Jim Martin, who's an ecologist in the biology department, and myself as a co-advisor. But he's become very central in this very interdisciplinary project. You'll see that reflected, I think, in his presentation, where he's been training both in ecology and genetics and bioinformatics, and also evolutionary biology, and trying to find how to use all those tools in this interdisciplinary project that he's going to describe. Noah also has really a great honor in that he received a NIFA fellowship, a pre-doctoral fellowship, a project that he wrote of his own on his own ideas to sort of expand on this project. And maybe he's going to mention a little about that as well. So with no further ado, Noah.
All right, can everyone hear me then?
Loud and clear.
All right, thanks, Mark. So I'm going to be talking today about, as Mark said, a portion of the Cacao NSF project, specifically focused on defense against, and the project specifically focused on defense against Phytophthora palmivera across 4 populations of cacao. So I'm going to start by giving a little bit of background on why we study cacao, why it's important, specifically why studying pathogens of cacao is important, as well as a little bit about the history of both the host and the pathogen. And then I'm going to talk about the NSF Cacao Genome Project, to give a little bit of background on that. And finally, I'll end with results from sort of preliminary results from our transcriptome project that we've done. I guess it was performed not really recently, but we've been analyzing the data somewhat recently. Okay. So why do we study cacao? Worldwide, production's about 3 to 4 million metric tons, which makes up a market that's between 90 and 100 billion dollars. So it's economically important. But even more than that, most of production, cacao production, centered around 5 to 6 million smallholder farmers, and many of which are in West Africa and Indonesia and South America, and actually constitute some of the poorest people in the world. So there's a huge room for economic development with the crop. And lastly, a huge, up to 40% of total yield is lost annually to a variety of different pathogens. So there's a lot of room for improvement. And these pathogens, a number of different pathogens are responsible for that loss. On the far left, here I'm just showing 3 of them. On the far left is frosty pod rot, which is caused by a fungus, Manilioptera rorii. It's particularly problematic in South and Central America. In the middle is black pod rot, which is caused by 2 Phytophthora pathogens, Phytophthora megakarya, and the pathogen I talk about today, Phytophthora palmivera. And then on the far right here is witch's broom, which is caused by another Manilioptera fungal pathogen, and is mainly problematic in South America. And actually, witch's broom has an interesting history because it represents sort of the first documented agrobioterrorism, where a disgruntled employee of the Brazil's USDA equivalent went around and tied infected branches to trees in these cocoa growers' plantations, and actually it resulted in sort of catastrophe for the cacao market in Brazil, but also the economy. It was hugely problematic. So it has sort of a sordid history. And we specifically focus on Phytophthora palmivera because it really represents the lion's share of annual losses. I'm showing here a number of different pathogens, the ones I showed you, as well as a few others, and their annual yield loss. And as we can see, black pod rot is at the top. And that's because it's particularly this black pod rot caused by Phytophthora, 2 Phytophthora pathogens. is problematic in West Africa, which is where about 70% of your cacao comes from. So worst pathogen in the highest growing area results in the most annual losses. And whether or not you realize it, you're probably familiar with Phytophthora pathogens, actually. They've been causing, they sort of have a long history of havoc. The most well-known example is Phytophthora infestans, which was the causal agent of the Irish potato famine. Here we see peasants lamenting the fact that their potato harvest is ruined in what was called the potato blight, or at that time it was called potato murrain. Phytophthora infestans is interesting though too, because it also represents the first described Phytophthora species. It was originally described in 1845 during the pandemic by a researcher. He was an amateur mycologist named Miles Joseph Berkeley, and he's and my perspective is he doesn't get enough credit actually, because he was the first one to sort of describe Phytophthora, and then to say, hey, what's causing the Irish potato famine? I think it's this lower fungus. We later realized that they're oomycetes, but at the time they're calling them fungi. This lower fungus called, well, he gave it a different name at the time. But often the person who gets credit is Heinrich Anton de Bary, who is a very well-known mycologist and was well-known at that time. He did the formal description of Phytophthora. And so he's often the one you see credited in books. But after his description in 1861, there were sort of a flurry, if you could say that, of Phytophthora species that are described. And presently we have 123 formally described Phytophthora species. And they have also been causing all sorts of problems. So a few well-known ones: Phytophthora capsicii is a pathogen of cucurbit species. Think squashes. Phytophthora ramorum is a pathogen of oaks and other woody plants. And then Phytophthora cinnamoni is pathogenic to hundreds of different hosts. And this really is representative of Phytophthora species, where they range from highly specialists, so Phytophthora megakarya, for instance, as far as we know, specializes on cacao, to pathogens that are have hundreds or even thousands of hosts. Phytophthora palmivora, the other. So these are the two agents of causal agents of black pod rot. One of them is a specialist and one of them is a huge generalist. And actually, these are images from Anton de Bary's original description of Phytophthora species. And I think they really elegantly show the infection strategy of the pathogen. So the pathogen first forms these, what are called these casings that are called sporangia. And these casings enclose the infection cells called zoospores. After certain developmental or environmental triggers, the sporangia release the zoospores into the wild, into the environment. And these zoospores are motile, and they move around in water via these flagella-like structures, where most of the time you think about bacteria pushing them along. Well, the Phytophthora zoospores are actually pulling themselves. So it's this one up here that's pulling itself through the water. Yeah. And so then after the zoospores find a host, what they do is they germinate. So I think this is shown really well over here on the right side. The aprosorium will land on a leaf. It will start germinating by forming an aprosorium. This is a little structure that helps it build pressure, and then it creates haustoria that either puncture the cells, the cuticle of the plant, or crawl through the stomata. I suppose unsurprisingly, plants have evolved ways to detect this. So here it's a bacteria that's infecting a plant, a bacterium, but it could very easily be a zoospore of a Phytophthora pathogen. And what these the plants have detected, the plants have evolved ways to detect presence of these pathogens extracellularly. There are receptors that sit up here that detect signals that are coming from the pathogen, as well as detect the intracellular things that are causing the infection. So these pathogens land, they'll secrete either secondary metabolites or proteins into the plant in order to subvert immunity. And plants have evolved ways to detect that as well. So once plants detect this, they launch a defense response that can take a number of different forms, some of which are formation of antimicrobial compounds, which I'll talk a little bit about today, as well as reactive oxygen species, and transcription factors that result in the transcriptional reprogramming of the plant. So potentially thousands of genes that can be upregulated to defend it. In recognition and subsequent defense response, we often think of as taking one of two potential categories. So defense in the plant can be qualitative, wherein there's a single gene or few genes that are conferring resistance. So a few dominant genes, and it's binary. It's either you are infected or you're not infected. Or it could be quantitative, wherein you see a spectrum of disease resistance and susceptibility. So no plant is either resistant or susceptible, but every plant is somewhere in between. And these different forms of resistance would appear differently if you were doing something like a QTL or determining quantitative trait loci or performing a genome-wide association study. Qualitative resistance would probably appear like a very sharp peak at a specific genomic position, whereas quantitative resistance would appear more like many areas of the genome being significantly correlated with disease resistance. Okay, so once the particular genomic positions that are important for disease resistance are identified, they can be incorporated into breeding programs, wherein a virus, say a virus-resistant plant, is crossed with a high-yielding crop. And then through subsequent generations of backcrossing and phenotyping, they come up with a plant that is both highly resistant and high yielding. The problem is both QTL mapping and then incorporating those QTLs into conventional breeding is it takes a long time. It works really well for things like corn and wheat. It doesn't work as well for things like perennial tree crops, which is what cacao is. Okay, so which brings us to the question of how do we identify genes that are conferring quantitative resistance in a long generation crop? And a specific way to sort of reframe that question is what aspects of cacao immunity are found across populations and represent sort of core pathways? And then how can we identify and functionally characterize those pathways? And this is the goal, was the main goal of the NSF Plant Genome Project. So here on the left, I'm showing you the PIs involved in the PIs and co-PIs involved in the project. Many are from Penn State as well as a number of other institutions. And the main goal was to first select a diverse set of genotypes from across multiple populations of cacao. Then with those genotypes, perform whole genome and transcriptome sequencing, and then use that whole genome and transcriptome sequencing data to perform evolutionary genetic analyses and transcriptome analyses to identify candidate genes that can be incorporated into functional characterization pipelines. And then lastly, after functional characterization, develop markers that could be given to breeders to incorporate into breeding programs. Okay, so cacao's native range is the Amazon basin. So to start, the NSF project selected 4 populations that are native to this region: Nene, Iquitos, Marañón, and Guyana. And then phenotyped those many individuals in each of those populations for resistance to Phytophthora palmivera. So the Phytophthora phenotyping looks something like this. And this work was done by a former postdoc in the lab, Drew Pfister, and a collaborator at CATIE, which is a germplasm collection in Costa Rica, and her name is Mariela Leandro Munoz. Okay, so the phenotyping looked like this, wherein the leaves were sampled from the trees, they were placed in petri dishes and challenged with pathogen. After 48 hours, the lesion size was measured. And as we can see, some plants have very small lesions after 48 hours, whereas others are completely dominated by infection. The results of that phenotyping can be seen here on the left. The y-axis represents the genotypes that were sampled, and the x-axis represents the average lesion radius. So moving from top to bottom, we see the most susceptible individual is GU123. And then the most resistant individual is Pound7. And as you can see, there are both resistant and susceptible individuals in each population. we can also see that this sort of gradation in defense response is characteristic of what I was showing before about quantitative resistance, that it's not a binary you are resistant or you are susceptible, but it's a spectrum of resistance to susceptibility. Okay. So from there, we took the 4 most resistant and the 4 most susceptible plants. We performed whole genome sequencing, and we also imported them from Costa Rica. So this is me here smiling in front of the imported plants after we had received them and we confirmed that they were growing well. After importing them, we created rooted cuttings that could be used for experimentation. We didn't want to directly experiment on the plants that we imported because if we killed them, we wouldn't have them for future studies. So we created 300 rooted cuttings, of which about 160 survived. And then we used those rooted cuttings to perform the transcriptome experiment, which is what I'm going to talk about mostly today. So what we did is we took those 160 cuttings and we distributed them out on 2 benches in the greenhouse, bench 1 and 2. And within each bench, there were multiple trays, A, B, and C. Okay, and so each tray would receive either a pathogen treatment or a control treatment. And the positions of the genotypes in each tray were randomized. So what we end up with is a kind of a confusing looking arrangement that looks something like this, but more or less it's a randomized block design, wherein say Scavena 6, which is a plant here, is treated with the pathogen. Well, its mirrored position on the opposite bench that would then be treated with water, and we do that across all benches and positions. And the final layout looked like this. And so from there, we infected the plants with agar plugs of Phytophthora palmivera on the underside of the plant, and collected the results at 8 hours. And we collected the outer ring a little bit outside of the pathogen lesion, here shown in red. So the final sort of transcriptome pipeline would, or workflow would look something like this. We imported the plants from Costa Rica, we created the rooted cuttings, we created the design of the experiment. We performed the experiment where we did infections collected at 8 hours post-inoculation on our 160 samples. We did RNA extraction, Lexogen sequencing, 3' sequencing on NextSeq 550, bioinformatic preprocessing, and then analyses. And of course, I didn't do this all alone. Much of this first part was done by Melanie Perryman or myself or both. And then much of this latter part was done by Eric Wafula or myself or both. So one of the issues working with a perennial tree crop is that you don't always end up with balance in the same way you would something like Arabidopsis. So we created, like I said, we created 300 rooted cuttings, of which about 160 survived. But from that 160, the distribution of individuals is hugely variable. So some genotypes, we only had 1 individual. Some genotypes, we had as many as 11. And even when we aggregate across the different genetic groups that we studied, we don't see an even number of individuals. And we don't even see an even number of resistant or susceptible. So the design is highly unbalanced, which takes special statistical considerations. So all through this process, design and then implementation of the differential expression models, we've been working with Dr. Naomi Altman, who's a professor emerita in statistics. Moreover, our collaborators said that greenhouses sort of give you the illusion of control over your experimental variables. The illusion is kind of right. There's actually quite a bit of variation even within a pretty confined space. So from bench 1A to 1B to 1C, so that's all the same table, different trays. We see large changes in both temperature and humidity. And we see that at both at time of inoculation and at time of collection. Okay, so we see huge amounts of variation in the environmental variables that have to then be incorporated into differential expression models so you can actually pull out signal. Moreover, it's similarly hard to control developmental variables. So when we take the rooted cuttings, they're all sort of at the same developmental stage, but as they grow to the point where we can do experimentation, their development becomes asynchronous from one another. So while we would have liked to do all the experimentation on a single leaf, that actually wasn't possible because just of the leaves that were available when we performed the experiment. And when you look at the expression results and you create a PCA, actually leaf developmental stage is most of the PC1. So most of the variance is coming from leaf developmental stage, which is to say that these need to be controlled for if we're going to actually pull out signal from our data set. So once we've controlled for all of these things, what we see is that actually treatment effects comprise the majority of our variation in the experiment. And by treatment effect, here's what I mean. This is sort of just a mock example where a given gene is responding to the treatment, but that response does not differ between resistant or susceptible individuals. We also tested for phenotype effects. Those are differences between resistant and susceptible, which would look something like this, where for a given gene, expression does not change from control to treatment, but there are baseline differences between resistant and susceptible that persist. And lastly, we can check for additive variation, which is shown here in the intersection of these two plots, intersection in this Venn diagram, where a given gene will respond to control and treatment, but there's baseline differences between resistant and susceptible classes that persists. Okay, so we can see here that about 1,600 of our genes are responding to treatment, which is far more than are responding to phenotype and additive effects. Okay, so one thing that we can do, that's sort of the first pass at these data, is we can create what are called volcano plots to essentially highlight maybe the most drastically changing genes. In volcano plots, so here I'm showing you on the x-axis is log2 fold change. That is log2 of the difference in expression between pathogen treatment and controls. And on the y-axis, I'm showing you negative log 10 of the p-value. So as you go up on the y-axis, actually the p-value is getting more significant. I've also color-coded the points according to whether or not they're significant, not significant, significant just by my log2 fold change cutoff, significant according to the p-value from the model, or significant according to both p-value and the log2 fold change cutoff, that's shown in red. You do see it, and so when a number of genes fall off in this upper right-hand category, meaning that their expression changes greatly from treatment to control, and that is highly significant in the model that we created, the differential expression model. But other than a few of these very highly changing genes, it's kind of difficult to parse through this, several thousand genes to find maybe specific functional pathways that are important for resistance. Okay, so one thing we can do is we can bin these genes, these differentially expressed genes, into functional categories. And from here on out, since as I said before, most of the variation in our experiment is coming from treatment, not phenotype. So I'm focusing mainly on the treatment effects from here on out. And when we bin those differentially expressed genes into functional categories, a few interesting things show up. So on the y-axis here are functional categories, or what are called gene ontology categories, that have significant enrichment in the data set. That is, there are more differentially expressed genes in this category than we would expect due to chance. So the categories are on the y-axis. Point size represents the number of genes in that category, and color represents the p-value of the enrichment. So, and then the x-axis represents the proportion of hits in that category. So, for instance, response to wounding, it looks like between 50 and 75 genes, and those 50 and 75 genes constitute a little less than 20% of that total category. And when we look at it, a number of interesting pathways show up, pathways that we more or less expected to see, like response to salicylic acid, response to jasmonic acid, response to ethylene, abscisic acid, and that's great. But there's a number of other pathways that show up that maybe we didn't initially expect to see and are therefore pretty interesting. and I highlighted those in red, phenylpropanoid and stilbenoid synthesis. And I'll come back to those, so keep those in mind. But Go categories, this sort of tells you potential categories that could be important, but it doesn't tell you maybe the magnitude of change within those categories. So to do that, I looked at the effect size of the given categories. So again, the y-axis represents those functional categories that are enriched in the differential expression results. And the x-axis represents the effect size, that is the log2 fold change, or the fold change, the difference in expression between the treatment and the control. So when you do that, what you end up with are, and then you rank them by the log fold change, we see that the phenylpropanoid and stilbenoid genes are responding to a greater degree than some of the other functional categories. So it sort of points to the fact that these two pathways could be important for disease resistance in cacao. What we can also do is we can just sort of, again, we're getting at ways to prioritize genes for functional characterization. And another thing that we can do with these differential expression data is we can correlate their expression, the expression of the functional category, with pathogen load. Here, what's nice about RNA-seq is that if you're using a pathogen that has polyadenylated mRNA, you actually get both pathogen reads as well as host reads. And so what you can do is you can take pathogen reads over the total host reads plus pathogen reads, total number of reads, to get proportion of pathogen load that are in your samples, so that you can then correlate that with the expression of these functional categories to see which categories are responding strongly to increased pathogen load. And so it would look something like this, where the x-axis is the log2 of the pathogen load, and the y-axis represents the mean normalized count for a given functional category. Colors, again, represent resistance versus susceptible, and shapes represent genetic group. Okay, so when we do this, so here, nitrite transport has a pretty loose correlation with pathogen load. But when we do this across all of the functional categories that are enriched, what we see is most of the categories actually have a positive correlation with pathogen load, which is to say that as pathogen load increases, the signal is getting stronger, and so these particular functional categories are responding more strongly in terms of their expression, mean expression. Okay. And finally, we can rank pathways according to effect size as well as their correlation with pathogen load to get an idea of maybe what are the most important pathways for this defense response. So that's what I've done here. I've ranked according to effect size in the treatment, as well as the correlation of pathogen load, so the correlation coefficients. And what I've ended up with is phenylpropanoid and stilbenoid, here labeled flavonoid biosynthesis, are the most important categories in this particular defense response. There are others, but these are the two that I'm narrowing in on for now. And this is interesting because polyphenolics that are coming from the phenylpropanoid pathway have demonstrated roles in pathogen resistance, either constitutive or phytoalexins that are turned on in response to the pathogen. The most basic phenol would look something like this, where you have a benzene ring attached to a hydroxyl group, and up to much more complicated polyphenolics that have multiple benzene rings. Here are two examples of known phytoalexins. So these are polyphenolics that respond, polyphenols coming from the phenylpropanoid pathway that would respond to a pathogen challenge. One is metacarpin from alfalfa, and the other is resveratrol from grape. And polyphenolics have demonstrated a demonstrated role in cacao resistance as well. So the phenylpropanoid pathway has been shown to be important previously. In work done by Ben Nolenberg, who's a former PhD student in the lab, now postdoc, he identified a compound, a polyphenolic compound called clovamide. And what he saw was that Clovamide has a much higher abundance in the resistant variety, Scavena 6, shown here in green, than in the susceptible variety, shown here in red. And when you take this compound and you put it on plates, agar plates, with different concentrations, you see decreased growth of Phytophthora pathogens, multiple Phytophthora pathogens, Megakarya, Palmivera, as well as Tropicalis. So the phenylpropanoid pathway has been highlighted before, and so it's also showing up in this data set, which is pretty interesting so far. Now, if looking at just the genes that are differentially expressed in the phenylpropanoid pathway doesn't give you a complete picture of how this pathway is potentially regulating defense response. So in order to do this, you can, to sort of determine what other genes may be involved with these differentially expressed genes in the phenylpropanoid pathway, I've created what are called co-expression networks. And co-expression networks are, I suppose, sort of just like what they sound like. It's genes that, genes that have highly correlated expression profiles. So what you can do is you can take all the expression of all of your genes, and then you can correlate their expression profiles. And what they'll do is they'll cluster into sort of hairballs that look like this, and then you can look at functional enrichment within each of those clusters. So here I'm showing sort of a mock example of what one of those networks, co-expression networks, could look like, wherein all of the genes in here have expression profiles that are very tightly correlated. And then if you look at the potential functional enrichment of these categories, we see, oh, many of these genes are probably involved in response to abscisic acid, or for example, or similarly, maybe this one down here, these genes are involved in response to drought stress. So it's a way to identify pathways of genes. The implicit assumption being that if two genes have correlated expression profiles, then they're most likely functionally related to one another. You can also use these co-expression networks to query genes to find sort of guilt by association. So here, if you say take a subset of that network, that sort of very complicated network I showed you before, what you can do is say, here's my gene of interest. What other genes are highly correlated with the expression of this gene? The width of, and here in this example, the width of these lines demonstrates the correlation of expression between two genes. So a thin line means these two genes are not highly correlated. A thick line means they are highly correlated. So what I did is I pulled the, so what I did is I pulled the genes that are differentially expressed in the phenylpropanoid pathway and used them as query genes, sort of bait. If you throw them into the network, what genes do you pull out that are highly correlated with those phenylpropanoid genes? And remember, I said that theoretically, the genes that are highly correlated with one another should share similar pathways. So when you use these phenylpropanoid, the differentially expressed phenylpropanoid genes as bait, what you pull out is the rest of the phenylpropanoid pathway. Okay, that's sort of a sanity check. Did we identify genes that are functionally related to one another? And we do, okay. But we also identify a number of genes that are highly correlated with the phenylpropanoid pathway, though they're not in that core phenylpropanoid pathway I just showed you. And what we see is that a number of the genes that have expression highly correlated with the phenylpropanoid pathway also have very well documented roles in defense response. For instance, a respiratory burst protein that's known to be involved in hypersensitive response. There are multiple Werke transcription factors that are very well characterized mediators of plant defense. There's a RIN4 protein, which is it is known to interact with certain receptors that confer resistance. And then a pleiotropic drug resistance protein, which is they're a class of transporters that are known to transport secondary metabolites. So what I'm trying to convince you, and hopefully is that the phenylpropanoid pathway seems to be important in cacao's defense response against Phytophthora palmivera. And working from about 1,600 genes that are differentially expressed, we can actually come down to fewer than 100 genes that seem to be highly correlated with pathogen load, are changing a lot in response to the pathogen, and have very well documented roles in defense response. Okay, so in conclusion, measuring environmental variables and developmental covariates is necessary to pull out signal from large and complex data sets like I was showing you before. Multiple genes, and then once we analyzed those data, we saw that multiple genes in the core phenylpropanoid pathway are differentially expressed, and their effect sizes and correlation with pathogen load suggest that they are significantly responding to pathogen treatment. And lastly, using these differentially expressed genes as queries into a larger co-expression network, we can retrieve not only the rest of the phenylpropanoid pathway, but we can also identify many other genes that are have well-characterized function and therefore might be good candidates for experimentation. In the future, I would like to further prioritize candidate genes using some of the integrating some of the population genetic analyses that we did or were done by our collaborators at Minnesota to see if any of the genes that are in this network that we've identified may be evolving differently in resistant and susceptible varieties. So to do that, we'd use what's called a population branch statistic, which I think last week's speaker talked about, but it's essentially a way to identify regions of the genome that are evolving differently in the resistant varieties than the susceptible varieties. So we could use that then as sort of another layer of evidence to identify candidate genes for functional experimentation. And with that, I would like to thank my committee members, as well as the whole NSF group and the multiple labs that I'm a part of, the Pamphilis lab and Gilton and Maximova lab specifically, as well as a lot of the work I was showing today was done by the people highlighted, myself and the people highlighted in bold here, Dr. Eric Wafula, Melanie Perryman, Dr. Tuomas Hamala, and Dr. Naomi Altman. And with that, I would love to take any questions if people have them.
Thanks very much, Noah. That was excellent. I see we did have one comment. It wasn't as much of a question as a comment that's in the chat window. Aaron Connolly commented that the phenylpropanoid pathway, PDRs and RBOH genes are also involved in plant iron metabolism, I guess. So yeah, it's amazing how projects seem to touch. So iron's well known, another part of the plant defense pathway. So
that's interesting because there's also a number of within that network I was showing. There's a number of cadmium responding genes, I think was the annotation. I don't know what that means necessarily, but metal response falls out within this network as well.
Yeah, one current trend is that iron chelation is one mechanism of plant defense response to withhold iron from, especially for bacteria. Are there any other questions? Let's get a few good questions for Noah. He did a lot of work. Here, let's get him, put him to the test.
I have a question for Noah. This is Ben Nolenberg.
Hey, Ben.
Great talk. Thank you for presenting. You were showing those sort of hypothetical plots for the different effects that you might observe for genes like treatment effect and phenotype effect, and then the additive effect. I didn't see like a crisscross pattern though. Like, have you found any genes that are sort of responding in that way?
Yeah, so like a phenotype by treatment interaction. No, we tested for it, but we didn't see anything. That's why I didn't include it here. That is, yeah, you're right. That is a potentially interesting category, but we think that our phenotype effect is probably a little too weak to detect the treatment by phenotype interaction.
Mhm.
So when we're looking globally at the data set, we don't see that. When we look individually at genetic groups, which I didn't show here, you actually do see some genes that are responding. But in that case, it's a genotype by phenotype interaction. But yeah,
Okay, thanks.
I'm sorry. It's still phenotype by treatment. I misspoke.
Okay. I got you. Thank you.
There's a message in the chat window from Dr. Martin. In your synthetic pathway slide, there's a tight cluster of DE genes on the right side. What are they?
One second, let me try and go back there. Are you talking about these ones over here? Yeah, I would need to look at those specifically. I'm not sure exactly what falls out. When I looked at these genes, I wasn't looking at position. I was just looking at annotations within this network, and I ranked them by log fold change. So actually, these are some of the most highly, the genes that are responding strongest to treatment. But I don't know. Yeah, I should look at what exactly is in specific positions.
Okay, this is pretty, we're still pretty early in this. This is really fresh data. That's you're seeing the first preliminary results, I would say. Even Noah's got a ways to go. Any more questions for Noah? Okay, well, if not, let's everybody thank Noah. That was a terrific seminar. Thank you so much, Noah.
Thanks, everyone.
Great job.
Nicely done, Noah. Really good.
Yeah.
Did the last network surprise you?
Yeah, I loved it.
I did those last night, so
Yeah.
I had other stuff finished, but then I got a little too
Yeah.
intrigued, and I started. Anyways, it morphed into